All right, next I'd like to begin our discussion of tumor suppressor genes. So let's draw our little schematic here of a gene and remind ourselves a few things about tumor suppressor genes. Remember, their job is to stop the cell cycle. And remember, we said before they are recessive, meaning both alleles have to be mutated. Unlike proto-oncogenes, where only one has to be mutated. Tumor suppressor genes we think of as both alleles having to be mutated. I also said before that mutations of tumor suppressor genes linked to cancer are non-functional. So just like I did with the proto-oncogenes, I'm going to ask a few questions to help drive this point home, because sometimes this is confusing, as I've experienced in previous semesters. If we made one of these genes, or both of the genes, overactive, would that lead to cancer? So if we cause this tumor suppressor gene to be expressed at a higher level in some way or another, is that going to help stimulate cancer? And the answer is no. Remember, the proteins that come from these tumor suppressor genes stop the cell cycle. If you make more of a tumor suppressor gene, you're just stopping the cell cycle even more effectively. It could be a problem, but it's not going to lead to cancer. So tumor suppressor genes that become overactive is not the link to cancer. A tumor suppressor gene that makes a protein that is inactive, so I'll just put an X there for being inactive. What will that do? So if you make a non-functional protein from this gene, so there's some kind of mutation here that leads to a non-functional tumor suppressor gene encoded protein here. Will that be non-functional? And I'm going to write here no if only one copy is mutated. If one copy is mutated, but the other copy is still making a wild type tumor suppressor protein, then it's going to be able to stop the cell cycle. This one can't stop the cell cycle, but it's not going to prevent this unmutated one from stopping the cell cycle. To stop the cell cycle, we only need one tumor suppressor gene to be present. But if both are mutated, both are going to produce a non-functional protein. If you don't have a protein being made that can stop the cell cycle, then the cell cycle will keep moving forward. So yes, if both copies are mutated. Or overactive mutations will not lead to cancer. Non-functional mutations will lead to cancer, but only if both copies are mutated. All right, now what I want to do is move to a specific example of tumor suppressor genes as we look at a pathway. And the proteins we're going to discuss here are RB, which stands for retinoblastoma, E2F, and P53, and P21. We're going to talk about a pathway that involves all of these proteins. Okay, so now let's consider how some of these proteins encoded by tumor suppressor genes function in regulating the cell cycle, and how when they are mutated, they can lead to disease, such as cancer. So let's draw a rather large cell here. And let's draw our nucleus over here. And in our nucleus, let's draw some DNA. And let's say that on our DNA, that we have a gene. And let's just call this an S phase gene. Meaning that this gene needs to be expressed in order for the cell to enter S phase. Until it's signaled to do so, though, this gene will be off. Let's also draw where a promoter might be for this gene, so that we can see that it is not activated. In order to activate it, there is this protein called E2F. E2F needs to enter into the nucleus and bind to the promoter to activate transcription. So E2F is a transcription factor. So let's just write TF under here, so we remember that it is a transcription factor. Now, we don't always want E2F in here, because if there are mutations present or the cell is not ready to enter S phase, we don't want this gene to be transcribed. So the way the cell prevents it from being transcribed is that another protein binds to E2F, and when it's bound to E2F, it cannot enter the nucleus. And this other protein is called RB. RB stands for retinoblastoma. Retinoblastoma is an eye cancer, and so it was first identified as a gene linked to eye cancer. However, mutations in RB are linked to other kinds of cancers as well. So before we continue the discussion, let's just draw our favorite cell cycle down here. And remember, we have this checkpoint here. And this checkpoint here won't allow a cell to enter S phase until it's ready to. And we said that the enzymes that help regulate whether or not the cell cycle will progress between these checkpoints are CDKs, cyclin-dependent kinases. So what happens here is that we have a CDK, a cyclin-dependent kinase. When the cell cycle is at this checkpoint, and we're not entering into S phase, this is how the cell looks. However, once the cell has assessed that there are no mutations, that the cell is the appropriate size, and other enzymes are ready in order to enter S phase, the signal is given to move forward. And that signal comes in the form of cyclin. When cyclin binds to CDK, it activates CDK. Once activated, CDK will phosphorylate retinoblastoma protein. So this active CDK cyclin phosphorylates retinoblastoma. When that happens, retinoblastoma undergoes a conformational change. So I'll write that up here, conformational change. once it's phosphorylated. Because of that conformational change, what happens is E2F is now released, only if retinoblastoma is phosphorylated. So it is now released, E2F is now free, and it can now enter into the nucleus, and it binds to this promoter. And once it's bound to the promoter, this S-phase gene is now activated. And now we can make this transcript and make the protein, and now this block is removed, and the cell can enter into S-phase. So this is what should happen. I also want to talk about something else that is important to happen. Remember, one of these checkpoints here is to check for DNA damage. If there's DNA damage, we don't want E2F coming in here. So how does the cell recognize that there's damage in the DNA to keep E2F bound to retinoblastoma? So let's generate some mutations on the DNA. So there are mutations here, and there's an enzyme that is going to recognize these mutations. And that enzyme is a very important enzyme, and it's called P53. P53 recognizes these mutations, and it does one of two things. It might fix the DNA, but sometimes, as we said before, if the damage is so severe, P53 will signal apoptosis and kill the cell. That's all we're going to say about apoptosis here, but just recognize that it is one of the pathways P53 can initiate. But let's talk about the fixing pathway. If P53 initiates the fixing pathway, the DNA repair pathway, in fact, let's call it specifically that, DNA repair. So if P53 initiates DNA repair, it will first do so by activating a protein called P21. If P21 is activated, what P21 will do is that it will inhibit cyclin-dependent kinases, and it will prevent cyclin from binding. So it inhibits CDK binding with cyclin. In doing that, the cyclin-dependent kinase is unable to phosphorylate retinoblastoma. If retinoblastoma is not phosphorylated, then it's going to keep E2F bound to it. If E2F is bound to retinoblastoma, E2F can't enter the nucleus. If E2F can't enter the nucleus, it cannot turn on these S-phase genes. And now the cell will remain stopped at the G1S checkpoint until this DNA damage is repaired. We're not going to talk about the pathway, the genetic pathway that leads to the repair of this, except to say that when P53 initiates the activation of P21, it also initiates pathways for DNA repair. Once the DNA has been repaired, P53 no longer activates P21, so these pathways are no longer activated. If P21 is not activated anymore, it is not able to inhibit the CDK-cyclin interaction, so now CDK can bind to cyclin. If it binds to cyclin, we will initiate the same pathway we talked about before, where it phosphorylates it, releasing E2F, so that E2F can now enter into the nucleus to turn on these S-phase genes. So it's a nice little pathway where retinoblastoma regulates whether or not E2F comes into the nucleus, and how P53 helps this cell recognize DNA damage and activating appropriate pathways to either fix or kill the cell. There are other pathways similar to this that we will see at G2M checkpoint and the spindle assembly checkpoint. We're just not going to talk about them in this class. As you're studying this, I recommend that you write this down, find a big whiteboard somewhere or chalkboard somewhere, write this out down, talk about exactly what I talked about with DNA mutations, which enzymes get activated. And then I want you to think about what happens in this process, in this pathway, if P53 is mutated, if P21 is mutated, if RB is mutated, if E2F is mutated. What happens? Or if CDK or cyclin is mutated. What happens if RB can no longer bind to E2F? Or what happens if RB is permanently bound to E2F because of some kind of a mutation? What is the result to the cell cycle? And will that lead to cancer or will it not lead to cancer? Those are the kinds of questions that will come at you on the exam, thinking about how these genes, if mutated, could lead to cancer or some other kinds of diseases. But that's all I have to say for now. If you have any questions at all, please make sure you come see me. If not, I will see you in class. Bye.